Hello, good day to everyone. My name is Matias Munoz and I am a PhD student at the Free University of Amsterdam. And today I'm going to talk about how environmental and morphological constraints can influence the evolution of frog calls. So whenever you see an animal producing a vocalization, you will observe that the production of the sound involves the movement of body parts, of body structures. So in these three examples, you can see three animals producing long-range vocal signals intended to communicate with distant receivers. And you can see all the sets of motions that are involved in these displays. So when I talk about environmental constraints, I am talking about any environmental feature that can somehow influence these processes of inflation, deflation, or body movement associated to vocalizating, and how these have an impact on the sound produced. And if you want to study how the microenvironment or the small-scale environment affects the production of sounds, then frogs are a very suitable group to study because they have this very conspicuous vocal sac that they inflate every time they call. And also frogs of different species use different calling sites. And I show you on this picture just the diversity of calling sites that you can find among frogs. So you can broadly put them into three categories: the ones that call from the water, the ones that call from the ground, from terrestrial calling sites, and those that call from vegetation, right? And for the matters of this work, we were interested in what are the constraints experienced by frogs that are inside the water versus the constraints imposed by the environment on frogs that call from outside the water. And the inspiration for this study comes from this studying this single frog, which is the Tungara frog. And if you look at it carefully, you will realize that a calling frog is no more than balloons connected by a larynx. So whenever the frogs push air out of the lungs into the vocal sac through the larynx, then the vocalization is produced. So if there is any environmental influence that can impact the process of inflation or deflation of the lungs or vocal sac, then we can expect to have an impact also on the sound that this frog produces. And this is the idea that Wouter, my supervisor, tested with these Tungara frogs. So he put them into an enclosure where he could manipulate the water level. And on the meanwhile, he was recording with a microphone. So he expected that if you have a floating frog, then it can inflate without constraints and will produce one type of call. But then if you decrease the water level of the same frog, then you have an on-floating frog, and then you record the call, it will be a different call. And it might be different because of changes in how the frog inflates these structures. So I will show you now a short video of the experiment. You will see first the frog in the lowest water level, and it will be doubled every time. So you can, one, listen to the calls at each dip, and you can also see how inflated is the vocal sac of the frog. So I will give play now so you can hear and listen. So when measured, what you find is that these are the box plots of the data obtained from this experiment. What you find is that vocal sac inflation is larger in floating frogs. They are unconstrained. And this is also accompanied by a decrease in call frequency. So floating frogs that can inflate completely will call at lower frequencies than the other males. And they will be more attractive to females also. So having this in mind, then we also, then focused on the different constraint, which is the morphological constraint, and there is no big mystery here. It's the tendency of larger animals to produce lower frequency sounds just because they have larger laryngeal structures. So this is a poster of frogs from Madagascar, and I put it here just because it shows the size of different frog species very nicely. So you can see this big on the center. We would expect this frog to call at lower frequencies than any other of the smaller species around it. This, for that matter, happens with all the vertebrates, basically. So it's a fundamental rule of acoustics. So with this in mind, we ask whether we can extrapolate these intraspecific ideas into multi-species comparisons. So we were wondering, can we find these lower frequency calls in aquatic species if we compare them with frogs that are calling from outside the water? So we did this studying 3 different families of frogs: the Ranidae, Leptodactylidae, and Hylidae. And for 160 species from these 3 families, we collected 3 types of data. We collected the body size, which is the snow vent length, SBL, the dominant frequency of their calls, the portion of the call with the highest energy, which is the F, and also whether these frogs called from the water, from outside the water, or they could be in both, which is the mixed category. So here you can see all the data that we collected from these 160 frog species. So the first thing we try is to relate the dominant frequency against the body size to try to find an effect of calling site. So what I will show you now is these are phylogenetic regression lines for the 3 families. You have on the y-axis the dominant frequency, on the x-axis the body size of the frogs. And what you see is that in the 3 families, aquatic species tend to have lower dominant frequencies than the non-aquatic species. We excluded the mixed category from this analysis, but I show you them on the plot anyway, so you can have an idea of where these species were located. The difference is not significant in Ranidae. It's a single intercept model, but it's significantly different for Leptodactylidae and Hylidae. So what we show here is that effectively aquatic species call at lower frequencies than non-aquatic frogs. We wanted to have a second look at this data using a different way of analyzing it, and we used this phylogenetic path analysis to identify causal links between these 3 variables: calling site, body size, and dominant frequency. So we tested 3 different hypotheses about how these 3 variables are related. The first one is the null hypothesis, because it only has the allometric association. Larger animals produce lower frequency sounds, and we know this across vertebrates. So this is the default model, right? The hypothesis 2 includes one direct link going from calling site to body size and still maintains the allometric arrow. And the third model, hypothesis 3, is that these 2 variables, body size and calling site, each one by separate has an influence on dominant frequency. So we ran the models, and then we found that when we analyze the 160 species all together, then it's a mixture of everything. In this figure, what you see in red color means that the coefficient of association between these variables is negative. The width of the arrow tells you something about the strength of the association, which goes from -1 to +1. And what we see is that in the end, the final average model includes all the possible arrows. There is a direct effect and positive direct effect of calling site on dominant frequency, but there is also the effect of calling site on body size, which indicates that there is some indirect effect on dominant frequency. So any effect of calling site on body size will have a concomitant effect on dominant frequency. So what we find with this path analysis is support to our idea that the frequency of the vocalization of frogs is under constraints by the immediate environment, and that aquatic frogs produce lower frequency calls because they are less constrained to inflate these structures than non-aquatic frogs. So what we are trying to do now in the incoming studies is to look deeper at these different calling sites and how they can influence the calls of frogs. So for example, we would be interested in have an idea of the timing of the changes across the phylogeny. So for example, are transitions from one calling site to the other rapidly followed by changes in the signals or not, or vice versa? Are changes in signals allowing animals to move to different environments? So we are trying to address these questions with a much larger sample of frogs, with a much more detailed acoustic characterization of their calls. And we are hoping to find many more interesting ideas about how the small-scale environment can influence the evolution of these mating vocalizations. And with that, I thank you all for your attention, and I will be very happy to receive and try to answer some of your questions. Thank you.